Good afternoon, everyone, and welcome to today's MNS webinar titled "The Neuroprotection Through Ion Channels: A New Pathway for Parkinson's Disease Treatment." It's a pleasure to introduce our speaker for today, Dr. Ines Bini, Associate Professor of Physiology and Neuroscience at the Pasteur Institute of Tunisia. Her research focuses on the mechanisms of Parkinson's disease and multiple sclerosis, and she's interested in studying the neuroprotective effects of animal venoms and plant extracts, and exploration of how ion channels are implicated in brain protection and brain disease recovery. Dr. Albini leads several national and international research projects, and she was recently awarded the James Gemini Award for her contributions to neurodegeneration research. She also serves as a North Africa representative on the Executive Council of the Society of Neuroscientists of Africa, SONA, and is an active member of the MNS Young Committee. Today's session will run for about 45 minutes, followed by a Q&A. And we will also share, by the end of the webinar, we will also share with you a survey link and an e-certificate link in the chat box for everyone interested in getting one. So without further ado, please join me in welcoming Dr. Ines Bini. Hello, Ines. Very pleased to have you today with us today. Yeah.
Thank you, Nermeen, and I would like to thank MNS for the invitation. I thank you for the introduction. So I'm Ines Bini. I'm Associate Professor at Pasteur Institute of Tunis, Tunisia. Also, I'm the head of the group, Neurosciences Group, and member of Biomolecules, Genome and Theranostic Applications Lab. So really, I'm delighted to be with you today, and I will talk about neuroprotection through ion channels. It is a new pathway for me for Parkinson's disease treatment. So I would like to start with a brief introduction about Pasteur Institute. Pasteur Institute is part of Pasteur Network, is a group of more than 33 research institutes around the world that work together to improve health and also science and innovation. The mission of Pasteur Institute is to protect and improve public health. It is main activities include the production of vaccines and sera, also medical diagnosis, biomedical research, and student training and education. I would like also to highlight something which is very important for us here at Pasteur Institute, that Dr. Charles, he was the director of Pasteur Institute, and he won the Nobel Prize for his work in typhus. It is very important for us to talk about Dr. Charles. Here we have 10 research lab, and I'm member of this lab, Biomolecules, Genome and Theranostic Applications. And recently, I recently established a research platform in experimental neuroscience, focused in research, neurosciences research. It combines basic studies on active biomolecules with applied research aimed to discover bioactive molecules that can slow or prevent neurodegeneration. And as you show here, so we use several types of approaches, from in silico, in vivo, and in vitro. What we have in our platform, we have in vitro and in vivo models, and also several molecular analysis tools, including Western blotting, immunohistochemistry, immunocytochemistry, PCR, and ELISA. And of course, all the team is composed by 100% of female. The atmosphere is lively, and the research from different, as you show here, master, engineering student, PhD, And we work together to understand the brain and to develop new treatment supported by our teamwork and also advanced equipment. It is a brief introduction about my institution here in Tunisia, my lab, and my team. So today I will talk about the implication or the involvement of potassium channels in the progression of Parkinson disease. And as you know, it is a neurodegenerative disorder that mainly affects older people. We study the role of ion channels in the development and progression of the disease in relation to the toxicity and its key protein, of course, alpha-synuclein. Let's start with Parkinson's disease. Parkinson is progressive neurological disorder. The hallmark of Parkinson include motor symptoms, as you know, and it is this motor symptoms is like tremor, rigidity, etc. However, several studies said that the presence of non-motor symptoms like depression, memory, loss of memory, etc. So this motor symptoms stem from the loss of producing dopamine. In addition, major facets of Parkinson pathology is related to alpha-synuclein aggregation, to microglia mediated neuroinflammation, and dopaminergic neurodegeneration. Of course, we have treatment. We have treatment strategies that aim to alleviate the disease symptom. But as you know also that we have only symptomatic therapy based on pharmacological, and this pharmacological treatment is based on the restoring dopamine level or mimicking its effect in the brain, but they do not stop or reverse the progressive loss of neurons that characterizes the disorder. Also, we have non-pharmacological treatment based on surgical, on physiotherapy, etc. So for Parkinson's disease, but why this pathology is difficult to treat? Because we have no diagnosis tests, we have no cure, and this pathology is complex and multifactorial disease. So for that, we should to try to answer the question, what is the cause of Parkinson? I really I like this slide, because for Parkinson it is a multiple pathologies. It is a multiple mechanism. That this is multiple targets. For that, when we to decide to work on Parkinson, we also try to understand this mechanism that can cause the pathology. Parkinson is caused by a complex interplay between multiple pathogenic mechanisms, and we know two competing theories for Parkinson. The first one is based on the loss of dopamine, which may be induced by mitochondrial dysfunction. But the second one is based on the formation of Lewy bodies. So today I will talk only about this second theory about Lewy pathology caused by misfolded form of alpha-synuclein. So alpha-synuclein is a brain protein, as you show here, which is physiological, which have a physiological role, which is very important for the regulation of the release of neurotransmitter, particularly dopamine. But we don't know why this protein can change the form and become misfolded and toxic, and the alpha-synuclein has the ability to spread from one cell to another cell via several pathways, transmission pathways, including direct penetration, also synaptic transmission, also via receptors. And in my presentation today, I will talk about some types of receptors, which is potassium channel. So what kills the question, what kills this neuron, the dopaminergic neuron? Really, it is a mystery, but there is a strong evidence suggesting that this is caused by the accumulation of this misfolded form of alpha-synuclein, as you show in this cartoon. From this point, my research hypothesis is based on this paper published by professor, my collaborator, Professor Hillel Lashwell. In this paper, in the conclusion of the paper, Hillel said that blocking receptors for extracellular alpha-synuclein, maybe it is the target to treat Parkinson. So, as I said, the propagation of this toxic form of alpha-synuclein can be done via several pathways, but this pathway via transmembrane receptor, which is very important for me. So the question is, are transmembrane receptors involved in the propagation of alpha-synuclein? And after that, we should characterize these transmembrane receptors. So this hypothesis, which the first point of to start this project, and the question is the involvement of potassium channel. Potassium channel are proteins that let potassium ion pass through cells. And we have several types of potassium channel, as you show in this cartoon. And one group of potassium channel called voltage-gated potassium channel opens and closes with changes after electric charge. And we can find this ion, well, this potassium channel in many places in the body, such as heart, kidney, and brain, and also potassium channel in the brain present these roles, as you show here. For example, cell volume changes, gene expression, calcium signaling, etc. But we don't know why. Maybe due to the mutation or exposure to toxins, these receptors can change and can cause diseases. And in this table, as you show in this table, we have some mutation in some types of potassium channel that can cause Parkinson's disease or Alzheimer or other types of neurodegenerative diseases. My question is, if we have some pathology or alteration in potassium channel, this alteration can cause Parkinson's disease. Exactly, Parkinson's disease, we can classify Parkinson's disease as a channelopathy. And in this slide, as you show, by this article published in 2015, when we have mutated gene or exposure to antibodies or toxins or abnormal transcription of normal gene, we can, after that, this pathophysiological alteration can cause alteration and can cause channelopathy and channelopathy related to diseases, neurodegenerative diseases, and of course, Parkinson's disease. So the question is Parkinson an ion channel disease or neural channelopathy? We'll try to answer this question, but we have not a lot of material to answer our question, because in the bibliography we have not a lot of paper to talk about this pathology like channelopathy. This one, for example, they said that this regulation in channels, and we talk general channels, together with abnormal calcium inflow in mitochondria and lysosome dysfunction, also with oxidative stress and toxic protein aggregation and neuroinflammation, can cause neurodegeneration in both sporadic and familial Parkinson's disease. And from this paper, we try to explore the effect and the function of potassium channel in Parkinson. And we are lucky because this work done by Sarkar and colleagues show that when we block Cav1.3 channel, we can protect neuron from neuroinflammation and neurodegeneration in experimental models of Parkinson. The Sarkar and colleagues used chemical molecules named PAP1, and from this paper this paper concludes that Cav1.3 upregulation in Parkinson models plays a major role in neuroinflammation mediated neurodegeneration. So via this work now we are sure that Cav1.3 is involved in the neurodegeneration in the experimental models of Parkinson's disease. Also, Sarkar showed that Cav1.3 is not elevated in microglia, but also in B cell lymphocyte, but this time in PD patients. So not only for experimental models, but also in patients when we have some upregulation of this channel. Also, in the same topic concerning potassium channel, a few studies, this 5 or more than seven studies, have explored the implication of potassium channel in the progression of Parkinson's disease through a number of experimental models, as you show in this slide. And these changes can cause the loss of dopaminergic neuron. So via these studies, we can conclude that both agonists or blockers of Cav channels could be used as therapeutic effect in Parkinson's disease. So from the literature, we can also conclude that targeting ion channel as therapeutic for treatment of Parkinson's disease is really a challenge, because in all studies, the research used only antibodies or molecules, chemical molecules, but for us, the originality of our study is to use molecules, not chemical or synthetic one, but from the nature. And we will show up after which types of molecules we will use in our studies. So how in this stage, our challenge is blocking alpha-synuclein aggregation and toxicity using molecules from animal venom. But why animal venom? because the first aim why we use animal venom, because a member of this lab which is created in 1987. In our lab, we have a library of biomolecules purified from animal venom. Also, these molecules were identified in pharmacological and structural characterization from scorpion and from viper. And also, we assessed the effect of these molecules in cancer and cardiovascular diseases. The molecules from animal, they have a high affinity, ample potency, and more specificity. Also, this choice was based on the existence of drugs extracted from animal venom that were approved by the FDA. For example, this one which is based from lizard, which is prevent the patient or to treat patient from diabetes. For this one, also extracted from viper and use it to treat high blood pressure. So we believe that some of these could be developed as drugs for Parkinson. Our first step was based on in silico analysis, and we selected compounds. So for the first time, we compare. The idea is to compare some molecules from venom, such as this one SHK from anemona. Say anemona, this one KH1 and KH2 from scorpion, and the in silico study helped us to identify the small library of molecules that can block Kv1.3 channel with more specificity and more affinity for this channel. So our goal is to find molecules that target Kv1.3, as I said, with more selectivity and more affinity. From scorpion venom, we identified KH1 and KH2 in collaboration with Professor Hillel Lashwell, and with the support of the center called Excellence in Africa at EPFL, École Polytechnique Fédérale de Lausanne, at Switzerland, and University Mohammed VI Polytechnic in Morocco. So for this project, the idea is to assess the effect of biomolecules from scorpion on our model of Parkinson's disease and to target Kv1.3 channel. Also, we have another project supported by Parkinson's Foundation and in collaboration with Professor Abuelezz at the Université Laval, Canada. And the idea is to try to assess the effect of biomolecules from anemona venom, which is called SHK and its analog. And I also, for this part of the project, we have another analog from SHK, which will be supported by national and local agency to try to block this channel and to found a drug for Parkinson's disease. Today, I will talk only about scorpion molecules and anemona venom, and I will start with this one. And we have this proof of concept KH1 and KH2 purified from venom of Tunisian scorpion, which is done by our colleague in the lab, Dr. Srairi. And these molecules can target two types of potassium channel, Kv1.1 and Kv1.3, but for our work, we aim to target only Kv1.3. The KH1 and KH2 have a special characteristic form called a functional diet, which includes two important amino acids. The basic one, amino acid, which usually lysine, and hydrophobic aromatic amino acid like tyrosine. And this functional diet is very important to specific recognition of target ion channel, and also to block the channel with more specificity. The first step in our study aimed to test two peptides, KH1 and KH2, from scorpion venom in primary cells, neuronal cells, looking at their effects before and after, as you show here, before and after treatment. And we aim to analyze the function by to determine the number of cells, also to check the number of pathological aggregates, which is our goal. And here, when we talk about aggregates, we talk about the aggregates of alpha-synuclein. In fact, this part was done in Lashwell lab at PFL, and as you show here in this slide, KH1 can block also the target of these molecules is Kv1.3 and can inhibit alpha-synuclein pathology formation, as you show in this part of the slide. And the number of aggregates decreased when we put at the same plate KH1 and PFF. PFF is the aggregates of alpha-synuclein. It is the same case for KH2, and also KH2 can block the channel, and it can increase the number of aggregates in our model. Both KH1 and KH2 show strong inhibition of alpha-synuclein seeding mediated pathology formation in a neuron, as you show in our results. And however, for this part of our study, KH1 was toxic in our cells. For that, we will try to continue the study using only KH2, because KH2 was not toxic in our cells. In fact, for more exploration, we treated alpha-synuclein knockout cells with KH2, with also the aggregates of alpha-synuclein, and the aim is to determine the level of expression of internalization of PFF. Biochemical analysis showed that at the concentration of 1 picomolar, KH2 decreased the level of fibrils internalization. Also, we showed that blocking Kv1.3 channels reduced neuroinflammation in cell lines, astrocyte cell lines, and this work was published in BBRC journal in collaboration with my student Noor Al-Hudaynayli. And when we induce inflammation in our astrocyte in this cell line, we showed that the expression of Kv1.3 increased in this inflammatory condition. But when we block the channel with molecules target this channel, the inflammation condition was restored. So from this part of our study, based on the assessment of the effect of biomolecules from scorpion venom and Parkinson disease models, the conclusion of this part said that both KH1 and KH2 show strong inhibition of alpha-synuclein seeding mediated pathology formation in neuron. Also, KH2 induces significant increase in glial cells, and KH1 is toxic. For the second part of this study, based on the to assess the effect of biomolecules from Anemona venom, and I talk about SHK and its analog. This project is supported by, as I said, by Parkinson's Foundation in the collaboration with Professor Abuelezz, and the idea is to assess the effect of our molecules in the model of LEPA model. This model is developed by Professor Wesleti, and LEPA model induces the formation of inclusions mimicking Lowy bodies in cell culture. And this work was done by Nur Hudaineli at the lab of Abid Wesleti. And for our results, the first step, of course, is to assess the toxicity of SHK and its analog. And we showed that the two peptides are not cytotoxic in N2A cells. For this part also, in the model of LEPA system, we showed that native form of SHK presented a more affinity and more effect, and to increase the number of aggregates approximately about 50% in these types of cells. Also, for the analog from this peptide, and this very low and potent concentration, as you show here, 150 picomolar, used to reduce the number of aggregates, and it is a very important result because at 150 picomolar, the analog is able to block Kv1.3 channel with high selectivity. Also, when we assess the effect of potassium channel blockers on optogenetic mediated alpha-synuclein aggregation in iPSC-derived neuron, the results was fantastic, and we can the results were validated in human iPSC-derived dopaminergic neuron, and as you show here, very little or no aggregates in the treated neuron versus the light treated positive control. The blocking the Kv1.3 channel modulated also ERK and AKT phosphorylation in line with the literature. This shows that our intervention inhibits pro-inflammatory and pro-synthetic signaling pathway via MAP, ERK, and PE3 AKT mTOR signaling pathway. The idea is to promote alpha-synuclein aggregation. So my conclusion is summarized in this slide. So Kv1.3 is like a glimmer of light. Cav1.3 contributes to the propagation of toxic alpha-synuclein, and when we block this channel, we can protect neuron from the toxicity of alpha-synuclein. We are still in the line of investigation of the signaling pathway involved in the internalization process. Also, we will try in the future to use human dopaminergic neuron of Parkinson pathology and neurodegeneration, and also we will try to validate all our results in in vivo models of alpha-synuclein toxicity and pathology formation. So to block Cav1.3 present a therapeutic alternative in early stage of disease, because peptide from venom can block the internalization of misfolded form of alpha-synuclein, and via our project we will try to present a new biomolecules with which is safe and which is more specific and present more affinity to our target. Really a deeper understanding of the molecular mechanism and signaling pathway involved in the internalization of the protein is needed. And now with my team, we are working on this in this part to identify and to highlight the cellular mechanism to for this neuroprotection, and how biomolecules from animal venom can protect cells from the toxicity of alpha-synuclein. So it is our challenge. So we try also to combine not only one molecules, but also other types of molecules, maybe chemical and native form of molecules, to block these channels and to present a new treatment of Parkinson's disease based on animal venom. By the end, I would like to thank all our supporting companies, of course, Excellence in Africa Center, EPFL, UM6P, also IBRO, Parkinson's Foundation, my collaborator, Professor Hilal Lashwell, Professor Abid Wisleti, and all my team, and more specific, my student, PhD student, Noor Hudaini. And thank you for your attention.